Hello. Please hit like button and subscribe my channel. Also press bell icon for future video notifications. Thanks.
University of Pittsburgh researchers have designed cancer-fighting nanoparticles that co-deliver a chemotherapy drug and a novel immunotherapy, according to a new Nature Nanotechnology study published today. The new immunotherapy approach silences a gene that the researchers discovered was involved in immunosuppression. When combined with an existing chemotherapy drug and packaged into tiny nanoparticles, the therapy shrunk tumors in mouse models of colon and pancreatic cancer. I'm excited about this research because it's highly translational. We don't know yet whether our approach works in patients, but our findings suggest that there is a lot of potential. Chemotherapy is a pillar of cancer treatment, but residual cancer cells can persist and cause tumor relapse. This process involves a lipid called phosphatidylserine, PS, which is usually found inside the tumor cell membrane's inner layer but migrates to the cell surface in response to chemotherapy drugs. On the surface, PS acts as an immunosuppressant, protecting remaining cancer cells from the immune system. In an independent study that was recently published in Cell Reports, Yi-Nan Gong, Ph.D., assistant professor of immunology at Pitt, also identified XKR8 as a novel therapeutic target to boost anti-tumor immune response. Like a ferry carrying people from one side of the river to the other, we wanted to develop a mechanism that allows nanoparticles to cross intact blood vessels without relying on holes, said Li. To develop such a ferry, the researchers decorated the surface of the nanoparticles with chondroitin sulfate and PEG. These compounds help the nanoparticles target tumors and avoid healthy tissue by binding to cell receptors common on both tumor blood vessels and tumor cells and prolonging the length of time they remain in the bloodstream. When injected into mice, about 10% of the nanoparticles made their way to their tumor, a significant improvement over most other nanocarrier platforms. A previous analysis of published research found that, on average, only 0.7% of nanoparticle doses reach their target. Next, the researchers tested their platform in mouse models of colon and pancreatic cancer. Animals treated with nanoparticles containing both Fu-Oxp and siRNA had better tumor microenvironments with more cancer-fighting T cells and fewer immunosuppressive regulatory T cells than animals that received placebo or Fu-Oxp doses. As a result, mice that received the siRNA Fu-Oxp nanoparticles showed a dramatic decrease in tumor size compared to animals that received those carrying just one therapy. According to Li, the study also pointed to the potential of combining the Fu-Oxp siRNA nanoparticles with another type of immunotherapy called checkpoint inhibitors. The researchers found that Fu-Oxp nanoparticles with or without siRNA increased PD-1 expression. But when they added a PD-1 inhibitor drug, the combination therapy had drastic improvements in tumor growth and survival in mice. With their sights set on translating their novel therapy to the clinic, the team is now looking to validate their findings with additional experiments and further evaluate potential side effects. Other researchers who contributed to this study were Yuang Chen, M.S., Yixian Huang, Ph.D., Qinj Li, M.S., Zhangyi Luo, B.S., Ziqin Zhang, M.S., Haojie Huang, M.S., Jingjing Sun, Ph.D., Lin Xin-Tian Zhang, B.S., Runzi Sun, Ph.D., Daniel J. Bain, Ph.D., James F. Conway, Ph.D. This work was supported by the National Institutes of Health, R01 California 219399, R01CA 223788 and R01CA 219716. 2022, November 25. Novel nanoparticles deliver innovative cancer chemoimmunotherapy. Science Daily. Retrieved November 28, 2022 from ift.tt/4s6 Psyche University of Pittsburgh. Thanks for watching. Please subscribe my channel for more videos.